Today I will be talking about the NK cells. So previously in 1970s, some cells showing spontaneous cytotoxic action was discovered, and ultimately Rolf Kiesling and his colleagues named them as the natural killer cells in 1975. So these natural killer cells were also known as the large granular lymphocytes, as they contain several cytotoxic granules. These NK cells are a part of innate immune system because they do not have any antigen specific receptors or antigen specificity. They represent the first line of defense against the viral infection and the tumor cells. And their one of the special property is that they can kill their target cells without any prior immunization or without the use of antibodies. Now this is a family tree of all blood cells. Starting from the hematopoietic stem cell, we can see that it produces the lymphoid progenitor cells, and it ultimately gives rise to the natural killer cell, B cells, and the T cells. Now the development of the NK cells. As we have already known that the NK cells are the descendants of the lymphoid progenitor cells. The maturation site of human NK cells are the bone marrow, lymph nodes, and thymus. Starting from the hematopoietic stem cell, it gives rise to the lymphoid primed multipotential progenitor cells. We can see the expression of CD34+, CD133+, and CD45RA+. These are the markers which are seen. When it produces the common lymphoid progenitors, in addition to the previous markers, we can also see the presence of CD38, CD10, CD127, and CD7 markers. So after the CLP stage, expression of CD122 marks the irreversible fate decision of CLP into a NK cell. And from the NK precursor cells, these immature cells rather, expression of CD56 marks the transition from the immature NK cells or the NK precursor cells to the mature NK cells. Here are some markers of NK cells listed in both the charts. So mainly this chart contains the CD markers, where we can see that the NK cell contains CD2, CD8, CD16, CD45, and CD56. The functions of each of the CDs or the cluster of differentiation molecules are listed in this table. Like the CD2 acts as addition molecule and signal transduction. CD5, the action is yet unknown, and CD16, it shows low affinity receptor for Fc region of immunoglobulin G. So the NK cells can also be differentiated from the T cell and B cells by the absence of T cell receptor and the B cell receptors. Apart from these CD markers, NK cell can also be identified by the presence of some receptors like the killer immunoglobulin like receptors, or in short KIR or KIR2DS1, KIR2DS3, etc. The NKP receptors, the LIR receptors, and NKG receptors. NK cell licensing. As we can see, this is a process done to maintain tolerance towards the self cells. Now, licensing is done by engaging the inhibiting receptors to recognize the MHC1 molecules. On proper interaction, NK cell will not kill the cells that show adequate amount of MHC1 signals. So this is done to give the NK cell property to distinguish between self and non-self cells based on the presence or absence of MHC1. If this process does not take place, then the NK cells might go on killing any cell to which it may interact. Now, how to identify the enemy cells? So as we already know that NK cells can kill any cell that does not show the class 1 MHC. So in short, it is called the missing self model, which was stated by Klass-Kerr, and it can be also stated as recognition of self MHC1 inhibits the ability to kill. NK cells express several number of receptors, but those receptors can be broadly categorized into two types, that is the inhibiting receptor and activating receptor. The inhibiting receptor is responsible for providing a negative signaling or an inhibiting signaling to leave the target cell unharmed, and the activating receptor, it provides a signaling to destroy or kill the target cell. As we can see in this diagram, in the normal cell over here, we see the presence of class 1 MHC, which interacts with an inhibitory receptor on the NK cell. It provides a no killing signal, so the normal cell is left unharmed. The inhibiting signals can override the activating signals, that is, can trump over the activating signals. As the cells express normal levels of unaltered MHC class 1 can escape all forms of NK cell mediated killing. In case of a viral infected cell where the MHC class 1 molecule is absent, we can see the activating receptor. It interacts with the ligand present on the virus infected cell and provides a signal to destroy the affected cell or the target cell present. Here are a few receptors which are listed in this table. Suppose the NKG2D. Now these receptors are mostly activating but not all. In case of NKG2D, it is a kind of activating receptor that when it binds to MICA or MICB in case of humans, but the NKG2A, when it binds to HLA in case of humans, it acts as an inhibiting receptor. Coming to the function, in case of antibody-dependent cell-mediated toxicity, that is ADCC, we see the involvement of antibodies. Now, it is known that NK cell cannot produce antibodies. So the antibodies produced by B cells, they can bind to the antigens present on the target cell surface. Now, these antibodies or the Fc regions of these antibodies are recognized by the NK cells, and those CD16 or the Fc receptors present on NK cell binds to these antibodies. Thus, the cross-linking of CD16 triggers the degranulation of the cytotoxic granules present in the NK cells into the lytic synapse. So, after the cytotoxic effect produced by the NK cells, the tumor cell or the target cell dies. So now for the apoptotic pathway, we see the presence of a Fas ligand which is present on the NK cell or the CD95 molecule, and the presence of Fas receptor which is present on the target cell. Now this procedure is same as the cytotoxic T lymphocyte action. So we'll see that on interaction of Fas to Fas-L, we see a downward signaling cascade that activates caspase 8, which ultimately cleaves BID protein to produce a truncated BID, which ultimately releases cytochrome C from mitochondria, and cytochrome C along with APOF1 and caspase 9, procaspase 9 produces caspase 9, which can cleave caspase 3 to initiate apoptosis. And caspase 8 can directly also cleave caspase 3 to initiate apoptosis. Now, in case of the perforin granzyme pathway, the NK cell containing perforin and granzyme in its vesicles, it is exocytosed. These granules are exocytosed and they are again internalized by the target cell by endocytosis. Now the granzyme B present in the endocytic vesicle is released inside the cell by a perforin mediated pathway. That is, the perforin forms kind of a pore within the vesicle or the endocytic vesicle, which allows the movement of granzyme B into the cell. So this granzyme B again acts to cleave BID and initiate a mitochondrial mediated apoptotic pathway involving caspase 9. It can also act to activate the procaspase 3, producing caspase 3 and initiating apoptosis. So the cell dies ultimately. Now this was all about the NK cell function, marker and development. I have listed some books for further studies and some papers to go through, and that was all. Thank you.